Hello everyone. Today we are going to start chapter number 10 Biotechnology and its application NCERT exemplar questions. Disclaimer. So what is going to be today's learning objective? That we will understand the fundamental concept of chapter number 10 Biotechnology and its application by working through NCERT exemplar questions. So today we are going to solve the NCERT exemplar question of this chapter, and by solving this chapter on all the way, that what we're going to do, we will understand its concepts, its fundamental concept and theory with the help of the questions which we are solving. So this is the index. So there will be like four types of question. One first will be multiple choice questions, which is from 1 to 10. There will be 10 multiple choice question. Then there will be very short answer type questions. Then there will be short answer type question, and at last there is long answer type question. And at the end of the today's session, we are going to complete 23 total questions and 23 different concepts from the chapter. Not different concept, but we are going to understand the concepts very better in a better way. So let's see the first question. So the first question says, this is a multiple choice question and this is from NCERT session 10.1. So first question is asking us, BT cotton is not, yeah. So question says that BT cotton is not, options are a GM plant, that a genetically modified plant, a insect resistant or a bacterial gene expressing system, or it is not resistant to all pests. So among these four options, what is something that BT cotton is not? So let's see the answer. So answer is option D, that is resistant to all pests. BT cotton is not what? Is not resistant to all pests. Why? Because BT cotton plant, it is first of all, it is a genetically modified plant. We have altered its gene. Its genome has been altered by the manipulation. And what is that manipulation? We have introduced the BT toxin gene into the plant. Now, this BT toxin is produced by which bacterium? So this BT toxin is produced by Bacillus thuringiensis, and this BT toxin gene has been cloned from the bacteria and is then expressed in the plants. And what is this providing? It is providing the plant resistant to some insect only. What are those insects? Those insects includes lepidopterans, that is tobacco worm or armyworm, coleopterans, which are beetle, and dipterans, which are flies and mosquitoes. Means it is not resistant to all pests. It is resistant to some of them. Among them, three classes are lepidopterans, coleopterans, and dipterans. So now let's go to second question. Now let's see what this second question is asking us. So the second question says, C peptide of human insulin is. So we know that the human insulin is consist of three peptide. Okay, initially three peptide. One is A, B, and C peptide. So this is from NCERT session 10.1, and the question is asking us that this C peptide of human insulin is what? So let's see the options. So the first option says that this C peptide of human insulin is a part of mature insulin molecule. Then second option says that it is responsible for the formation of disulfide bridges. Third option says that it has been removed during maturation of proinsulin to insulin. And fourth, that it is responsible for its biological activity. So let's see what the answer is. So the answer is answer C, that the C peptide in human insulin is removed during maturation of proinsulin to insulin, because we know that the mature insulin of human is consist of only two chain, that is chain A and chain B, which are joined together by disulfide bridges. This chain C peptide has to removed from that proinsulin, the first structure which is in the form of proinsulin, which has these all three peptides, to form into a mature insulin. Let's see the solution. So the solution says that it is an extra portion. This C peptide is of 31 amino acid in human proinsulin. So as we say, second, what was that? That C peptide has to be removed for what? For the maturation of this proinsulin into insulin. As I said, the proinsulin is in the form of three peptides A, B, and C. So to make it into a mature insulin, we have to do what? We have to remove that. Okay, from those three chains, we have to remove C chain, and then only it can became the normal insulin. Then this mature functional insulin is what? It is devoid of C peptide. Next question. So this is also a multiple choice question. Okay, and the question says us GEAC stands for, this is from NCERT session 10.4. They have given us a abbreviation and asking us the full form of that abbreviation. Option first is Genome Engineering Action Committee, B is Ground Environment Action Committee, C is Genetic Engineering Approval Committee, or D is Genetic and Environment Approval Committee. Let's see its answer. So the answer says that it is C, that is Genetic Engineering Approval Committee. What is this Genetic Engineering Approval Committee? It is the Indian government has set up this organization to make decision about any genetic engineering things, proposal, and what any genetic engineering research that has been happening. So these decisions are regarding the validity of genetically modified research to ensure the safety of introducing genetically modified organisms for public services. So on what decision means, they have been set up for making decision against what? So regarding what? So first they have to validate that this genetically modifying research, whatever modification we are doing to any organism, any crops, anything, is it validated or not? Is it good for society or not? Is it safe to introduce this GM, that is genetically modified organism, in the market for public services? So this is their duty. They ensures that the GM research, whatever is happening, it should be valid and it is like safe for public use also, public services also. So this is the function of Genetic Engineering Approval Committee. You guys should remember this abbreviation, this full form of the abbreviation. Next question. This is from this is also a multiple choice question. Question says choose the correct option regarding retrovirus. They have given us 4 options, okay, and they are saying us we have to choose one of the option which is true, which is correct regarding whom? Regarding retrovirus. This is from NCERT session 10.2.2, and options are first. They are saying that retrovirus are what? Is a RNA virus that can synthesize DNA during infection. Means they are RNA virus, means their genetic material is RNA, but they can synthesize DNA during infection. Means when they infect anything, infect human, they can synthesize DNA. Second says that it has a DNA virus that can synthesize RNA during infection. Third is a single stranded DNA virus, or the fourth is a double stranded RNA virus. So let's see what is the correct option. So the correct option is that it is a retrovirus, a RNA virus that can synthesize DNA during infection. So what is a retrovirus? It is a it has single stranded RNA virus. It stores its nucleic acid in the form of mRNA genome, that is from 5' and 3' poly A tail. Okay. In retrovirus, their RNA is reverse transcribed into DNA inside host cell only, and that is also reason it is called retrovirus. Means why? Because their RNA, its genome material which is of RNA, it can be reversed reverse transcribed, because we know that DNA is transcribed into RNA, but in this the RNA is reverse transcribed into DNA inside host cell only, means inside human only. So this is the picture of one of the virus, that is they are showing the human immunodeficiency virus. Okay, so you can see the picture. What is this? This is the RNA strand. Okay, single stranded RNA strand. This is the capsid which is protecting what is capsid? So capsid is the protein that protects the genetic material. Okay, then there is enzyme, there is envelope, and then on this envelope there are some envelope protein. So now we got to know what is a retrovirus and how it works. It is a RNA virus, means it has a genome of RNA, it has single stranded RNA, but during infection, what happens? It can reverse transcribed into DNA, and this can only happen when they are inside the host cell. So next question, this is also a multiple choice question, NCERT Exemplar 5. So the question says a protoxin is a very simple question. They are asking us what is a protoxin is. This is from NCERT session 10.1, and option says that first it is a primitive toxin, then second it is a denatured toxin, third it is toxin produced by protozoa, or it is an last option inactive toxin. Okay, primitive toxin means they are at very initial stage, like primitive, very simple toxin. Second, say that it is denatured, it got denatured, it is a denatured toxin. Third is saying that this protoxin is usually produced by protozoa. Or last says that this protoxin is inactive toxin, means this toxin is not activated. Let's see the answer. Answer is D, that is, it is an inactive toxin. We have studied that this protoxin is an inactive toxin, and this pro, actually, if this pro word signifies what? It is signifying its inactive form. Okay, like in insulin, we have studied pro insulin. Why it was pro insulin? Because it was not activated, it is not matured. Once it got matured, pro has been removed from it and it became insulin. Just like that, this pro here is signifying inactive form. This pro toxin has to get activated. How it can get activated? So we will see. First, see what is the third line, that B-thyrogenesis forms protein crystal is which in which contain a toxic insecticidal protein. Means we know that this uh, toxin is produced by B-thyrogenesis, and why and how it is in there. So it has a protein crystal inside it, and this protein crystal contains this toxic insecticidal protein, and this insecticidal protein, which is PT toxin, is inactivated. How it can get activated? It can get activated when it gets an alkaline pH. Alkaline pH means it has more basic environment. So, and where it can get this alkaline pH? So it gets its alkaline pH in gut of insects. Okay, let's see. First, see, as I have told that this toxin, this protoxin is, we get it from the Bacillus thuringiensis, and you can see that there is a protein crystal in which there is this Bt toxin. This has shown in the diagram. Other things are cell membrane, cell wall, DNA, endospore, ribosomes. But here, what is interest of ours, that there is a protein crystal inside which there is a Bt toxin. Now, next question is kind of a continuation of that question only, because last question has asked us what is this protoxin. So we have said that it is an inactive toxin, means it has not been activated. So now the question is, this is also a multiple choice question. The question says the trigger for activation of toxin of Bacillus thuringiensis is, this is from NCERT session 10.1, means what is the trigger, what is the point, what is the stimulus by which this protoxin, which is inactivated, got activated. So options are acidic pH of stomach, high temperature, alkaline pH of gut, or mechanical action in the insect gut. Let's see the answer. So The answer is, as I said in previous question also, that it is alkaline pH of the gut which is responsible of making this protoxin into toxin. Beta toxins are initially inactive protoxins. After ingestion by the insect, these inactive toxin becomes activated. This activation is due to what? It is due to the alkaline pH of the gut of insects which are consuming it. So this alkaline environment solubilize the crystal, activating them. This beta toxin, as I said, is inside a protein crystal. Okay. So uh, this protein crystal is like has to dissolve. So this alkaline environment is responsible for solubilizing that crystal. Once that crystal has been solubilized, what happened? Now this protoxin activate and became toxin. Okay, let's see next point. Under high temperature and acidic pH, Bt toxin remains insoluble and inactive. Means if you will give high temperature, if you will give high temperature and acidic pH, this Bt toxin will remain insoluble and it will be inactivated only. Now, next question. So what is next question? So next question is also a multiple choice question. This is NCERT Exemplar 7. This question says golden rice is. Now it is asking what golden rice is, and this is from NCERT session 10.1. So let's see the option. So options are: first, golden rice is a variety of rice grown along the Yellow River in China. Okay, so they are saying that golden rice is a variety of rice which we grow alongside with in China alongside with Yellow River. Okay. Second says it is a long store rice having yellow color tint. Means this rice has been stored for so long and it has what? It has a yellow tint. Means it has a yellow color in it. Third says it is a transgenic rice having gene for beta carotene. Third says it is a transgenic rice. Transgenic rice means it's genetically modified rice, and it is having which gene? It is having beta carotene gene. Whereas the fourth option says that this is a wild variety of rice with yellow colored grains. Okay, let's see the answer. So answer is C. This had that it's a transgenic rice having gene for carotene B, beta carotene. So golden rice is what? It is a genetically modified crops with enhanced nutritional value. It is rich in vitamin A. Okay, and it contains beta carotene gene from daffodil plant and also genes from some bacteria. So they have told us, like, what they're telling us, they have told us that golden rice is what? It is a genetically modified crops, and it is like why it has been genetically modified, because it has been genetically modified to enhance the nutritional value of this rice. And what nutrition is that? It has been rich in vitamin A. Okay, and it has this beta carotene in it, and this is also reason that it is golden in color. And this gene, the beta carotene gene, has been derived from which plant? So this is derived from daffodil plant or some other genes from bacteria. Okay, so next question is also a multiple choice question, and in this question there is asking, in RNA interference, genes are silenced using. Means in RNA, that is RNA interference, genes are silenced. We know that genes are silenced. How genes are silenced? So genes are silenced. Now they have given us four options. So first option says it is gene can be silenced using single stranded DNA. B, it has been silenced using double stranded DNA. C, they are saying that they have been silenced because of double stranded RNA. And last, they are saying that they have been silenced using single stranded RNA. Let's see the answer for this. So the answer is C, double-stranded RNA. So this RNAi, that is the RNA interference, it is a natural biological process. It involves inhibition of gene expression by silencing targeted mRNA. This specific mRNA is silenced due to complementary double-stranded RNA. So what is this RNAi? It is a natural biological process which happens in almost all eukaryotes. It involves what? It involves the inhibition of gene expression. So, and how it has been inhibit the gene expression? It inhibits the gene expression by silencing targeted mRNA. Means the target mRNA, okay, that this double-stranded RNA go, the complementary double-stranded RNA go and bind to mRNA and thus stop the translation. And this is how it works. This RNAi process is what? The double-stranded RNA contains a sense and antisense strand from for mRNA. It goes inside the cells, process inside the cells, and only complementary strand to the mRNA binds to it, inhibiting the gene expression. So the mRNA strand, which is the double-stranded RNA, which is complementary strand to mRNA, goes and bind to it, and thus it inhibits the gene expression. So next question says, it's also a multiple choice question. This question says the first clinical gene therapy was done for the treatment of, means the first gene therapy which happens, it happens for which disease? So this is from NCERT session 10.2.2. Options are AIDS, cancer, cystic fibrosis, or last SCID, that is severe combined immunodeficiency resulting from the deficiency of ADA, that is adenosine deaminase. So the answer is option D, that is SCID. The first clinical gene therapy was done for the treatment of SCID, that is severe combined immunodeficiency, which is resulting, why it happens? It's result from the deficiency of enzyme ADA, that is adenosine deaminase. So this SCID is disorder which is related to immune system. It is resulting from the deficiency of enzyme ADA, that is adenosine deaminase. The SCID patient have a defective gene for that enzyme. As a result, he or she lacks functional T lymphocytes. So this first clinical gene therapy has been done on a 4-year-old girl, okay, who has this what? Who has defective gene, the gene that is encoding for the enzyme ADA, that is adenosine deaminase. So what happen if you to have deficiency of this ADA enzyme, you can lead to, that person can lead to this SCID disease, that is severe combined immunodeficiency. It is a disorder which is related to immune system. Because of this enzyme, immune system is able to function properly, but if there has been deficiency of this enzyme, so the immune system will not work properly. So as a result, that person is going to lack the functional T lymphocytes. Next question, it is also a multiple choice question, and this question is also kind of like related to the previous question. So the question says that ADA is an enzyme which is deficient in a genetic disorder SCID. Okay, so what is the full form of ADA? We have seen in the last example question also. So this is from NCERT session 10.2.2, and the options are: first, adenosine deoxyamynase; B, adenosine deaminase; C, aspartate deaminase; or fourth last, arginine deaminase. So the answer is B, adenosine deaminase. So it is an enzyme which is deficient in genetic disorder SCID. Okay, and this enzyme is crucial for what? Immune system function. So we have studied this in previous question also, that if a person has a deficiency of this ADA enzyme in the body, it is going to lead to a disorder which is SCID, that is severe combined immunodeficiency, and this enzyme is what? Is very crucial and responsible for immune system to function. Now after this MCQ, we'll come to what? We'll come to this very short answer type question. So this is NCERT Exemplar 1, very short answer type question. So let's see what question is asking us. So the question is saying, in view of the current food crisis, it is said that we need another green revolution. Highlight the major limitation or the earlier green revolution. This is from NCERT session 10.1. So we have studied about green revolution in this chapter. So this question is basically asking us, even in today's scenario, we are suffering from kind of food. If there is like food crisis, shortage of food, so why there is a need of another green revolution? And if we need another green revolution, what was the limitation, what was the drawback of the earlier, of the previous green revolution? So the answer is, the answer is, what is the main drawbacks of the earlier green revolution? First, it generally and desired character also breed along with desired character, means we know that we are not using this genetically modified thing, all those genetic engineering into this previous green revolution. So what used to happen, we can get undesired character also with the desired ones. Then there has been the use of fertilizers which can affect the soil fertility and quality. This fertilizer, pesticides, okay, this is very harmful for soil. Extensive use of these pesticides, insecticides, and weedicides resulted in harmful effect on the ecosystem. As I said, this fertilizer, pesticides, insecticides, weedicides, they are not very good for soil and even causing harmful effect on the ecosystem. So another green revolution that can cure these problems and enhance the food quality food production in limited land resources certainly required, and that's the reason that we need another green revolution. So now, next question is also a very short answer type question. The question says, give the full form of ELISA. Okay. And which disease can be detected using it? Discuss the principle underlying the test. This is from NCERT session 10.2.3. First, they are asking us the full form of ELISA. Okay. Then they are asking that which disease can this ELISA can detect. Third, it discuss the principle which underlying the test. That and they are asking us that we have to tell what is the principle of this ELISA test. So the full form of ELISA is enzyme linked immunosorbent assay. Which disease it can detect? It can detect HIV, which is AIDS disease caused by HIV. And what is the principle behind this ELISA? So it is based on the simple principle which is the antigen and antibody reaction interaction. So as you guys can see in the figure, what can happen, how ELISA can detect it. For example, here you can see this is a capture antibody. Okay, this capture antibody is against which, what this capture antibody is against this target antigen. So this capture antibody can come and this antigen is going to interact with it because they are both specific. This antigen is specific to this antibody. Then the other detection antibody which is being labeled with enzyme. Which enzyme? The enzyme which has the power to convert the substrate into some, means it can convert the chromogenic substrate, means this enzyme has power that it will convert the substrate and the substrate will give us some color. So by that color we can understand, okay, so this has been against that specific antigen only. So that's how we can, I don't know, like this, we can detect the disease. For example, this target antigen would be of AIDS. Okay. So now, and we have captured antibody against this AIDS disease. So this captured antibody going to interact with this target antigen, and now this detection antibody which has enzyme with it, the star shows enzyme. It what is going to do? It is going to convert the substrate into some chromogenic product, and this chromogenic product means a color product will tell us, okay, it means there has been presence of this specific antigen of AIDS. This is how it works. Infections can be detected in two ways. by detecting the presence of antigen, which could be protein, glycoprotein, or by detecting the antibodies synthesized by the host against the pathogen. So, how we can detect this infection? So we can detect this infection with the by detecting the presence of antigen. As I told, this has been shown that we can detect this by the presence of the antigen. Here it's a AIDS disease, and AIDS disease we can know happens due to the HIV virus. Okay, so if there has been presence of any glycoproteins of HIV, any proteins of HIV virus, so we can detect the presence of that virus. Now, first we can either detect the antigen, and second we can either even detect the antibodies synthesized by the host against the pathogen. For example, if someone is infected with some infection, so that once that bacteria or virus goes into inside to human, it is going to produce some antibodies against that virus or any pathogen. Okay, so we can detect that antibody also, and by the presence of that antibody we can say, okay, so there has been presence of that pathogen inside the host. So this is the two way we can identify the infection. Now next question says, many proteins are secreted in their inactive form. This is also true to many toxic proteins produced by microorganisms. Explain how the mechanism is useful for the organism producing the toxin. This is from NCERT session 10.1. So, this question is asking us, there has been many proteins, okay, which have been secreted in their inactive form. One example we have seen of this PT toxin. First is what? They are in its inactive form. So they are saying, this question is saying that there are many proteins when they are secreted, they are in their inactive form. So this is also true when for many toxic proteins also produced by microorganism. So we have to explain how the mechanism is useful for organism producing the toxin. So why it is useful that that toxin is in inactive form for the organism who is producing that toxin. And this is from NCERT session 10.1 only. So, many proteins, including certain toxin, are secreted in their inactive form. So, and this toxin is called what? This inactive form is called what? Protoxin. Okay. What is the significance of this mechanism? So, the significance of this mechanism is that, first of all, this mechanism is advantageous to the toxin producing organism, example bacteria. It's saying that if a, as I said, that bt toxin is produced by bacterium. If the toxic is harmful to its own host, to the own bacteria, what will happen? That bacteria will die. So this is present in the inactive form in the bacteria, as it's useful to that bacteria. Okay, now second, this protoxin can only get activated when they're exposed to some specific trigger. In Bt toxin, it was alkaline pH, but it could be some temperature and any other thing also. Then third, the bacteria does not kill by the toxin produced inside it, as I told, because it is an inactive form. Okay, and it required some specific trigger, that could be any pH or temperature, to get activated. So it is good for that bacteria only, because it is not going to kill that bacteria who is producing it. Next question. This is NCERT example 6 question of very short answer type question. So now this is saying what? It is asking us, give any two reasons why the patent on basmati should not have gone to an American company. This is from NCERT session 10.4. So this question is asking us, we have to give them two reasons that why this patent which has been given to American company should not have been given to that company. Why? So this should be not be that patent should not be given to American company on basmati. Why? First, the basmati rice variety have been grown in India since time immemorial. Means it has been grown in India from centuries, and it is traditionally it belongs to whom? It belongs to India. The new variety of basmati that got patent rights to an American company is actually derived from Indian farmer variety. So what they did, they took a Indian farmer variety basmati and crossbreed it with a semi-dwarf variety and says, okay, now this is a new discovery, there is a novelty, we have produced a new variety of basmati and now we got that patent. Although it is not correct because it is derived from Indian farmer variety only and it belongs to India only. So that's why the patent should not be gone to that American company on basmati rice. Next question, it is also a very short answer type question, and the question says that how was insulin obtained before the advent, means discovery, of R, that is recombinant DNA technology? What were the problems encountered? Which is from NCERT session 10.2.1. So that this question says us, like, before we have like introduced this recombinant DNA technology, the scientists have introduced this recombinant DNA technology to the world, we have to obtain insulin from some way, okay, but why there was problem in that, why we can't take that insulin from that way which we are using before this recombinant DNA technology thing? So let's see the answer. So insulin is used for whom? They are used for diabetic patient and diabetes treatment. It was earlier extracted from the pancreas of slaughtered cattle and pigs. Means we have to kill those cattle and pigs and then we extract this insulin from the pancreas of them. Now, what is the problem associated with this insulin extracted for animals? First, we have to slaughter them. This is one of the biggest problem. Second, this insulin can cause some patient to develop allergy. Some people can have this tendency to develop allergy against this insulin because this they can treat it as foreign particles, and if they are treating it as foreign particles, it can going to trigger some immune reactions in some patient against that insulin. So these are the major drawbacks of using the insulin before the discovery of advent of recombinant DNA technology. Now, next question. Next question is also a very short answer type question. This is NCERT Exemplar 11, very short answer type question. And question says that we have to simply discuss the advantages of GMO, that is genetically modified organism. This is from NCERT session 10.1. Let's see what are the advantages of the genetically modified organism. So the organisms whose whose genetic materials have been altered by manipulation is what we call genetically modified organisms. So this genetically modified organisms can be either plants or animals. So this is a figure showing a genetically modified plant crop, that is rice, where one side it is normal rice and other side is the golden rice. And we know that we have genetically modified it for the nutritional enrichment, and which nutrient? That is the vitamin A. So now what are the advantages of GMO? When we are talking about GM crops, that is genetically modified crops, it can have shorter growing cycles, it has greater resistance to both pests, that is insects, and disease. It has higher yield and higher nutrition value, as we talked about vitamin A enriched rice, that is golden rice. Means what? If you're talking about what the advantages we are getting from this genetically modified crops, that first of all, they can be grown in shorter cycles, okay, they are resistant to many insects and disease. If we are talking about the bt cotton, bt corn, everything which is resistant to many insect, okay, because of this bt toxin gene, so we know that, so they are resistant to some like insect and disease, and then we can get higher yield, more quantity, higher nutritional value. For example, this golden rice we are talking about, which is enriched in vitamin A. So this is the advantages of GM crops. Let's see some advantages of genetically modified animals. So this genetically modified animals can also have increased production and nutritive values. For example, we have studied about this transgenic cow named Rosie, which may produce more nutritious milk. We know that this transgenic transgenic cow Rosie, what she can do? So she can produce this alpha lactalbumin, Okay, so this is a protein, this is a protein, and it's like that. So she can produce that protein like 2.4 gram of that protein in 1 liter of her milk, and it is very nutritional to infant. And this is how we can use this genetically modify animals for increased production and nutritive values of products. So now we come to short answer type question, and this is the short answer type question of NCERT Exemplar 1. And this is the question says that gene expression can be controlled with the help of RNA. Explain the method with an example. This is from NCERT session 10.1. So the question is simply saying this, like gene expression, we can control the gene expression with the help of RNA, and we have to now explain the method with examples. So gene expression can be controlled with the help of RNA. One of the technique is called what? RNA interface and interference and the RNAi. What is the mode of action of this RNA interference? That first of all, RNA interference involves a double-stranded RNA. It contains both sense and antisense strand for mRNA. It is processed inside the cell, and the only complementary strands to that RNA go and binds to it and inhibits the translation and inhibits the gene expression of that. For example, one of the example is the prevention of nematode, that is the Molontigine ecognitia. So this nematode is responsible for infects the root of tobacco plants. Okay, which do what? Which reduce the production of tobacco plants. So what we can do? We can prevent it by using RNA interference process. So we can use agrobacterium vector. So we know that the agrobacterium vector has this TI plasmid which can go into tobacco plant and cause tumor and all those things. We have studied it in previous chapter, biotechnology principle and processes chapter. So we can use this agrobacterium vector, and we can introduce the nematode specific genes into the host plants. Okay, so this gene would do what? It will go inside this tobacco plant. It will produce both sense and antisense RNA in host cells, and these two RNAs are complementary to each other and are double stranded RNA. Now what will happen? This initiates the RNA interference and hence silence the specific mRNA of the nematode. Now what will happen? This double stranded RNA, the complementary part of strand of that RNA, go and binds to the mRNA, specific mRNA of the nematode, and thus it initiates this RNA interference and silence the specific mRNA of the nematode, and the nematode will not be able to infect the plant. The parasite cannot survive in transgenic host and so prevents the plant from pest. As the complementary strand will go and bind to the mRNA, translation is stopped. So now there is no survival of this nematode in the transgenic host. So thus we can prevent the plant from the pest. Next question. So the next question says that we have to highlight any 4 areas where genetic modification of plant has been useful. This is from NCERT session 10.1. So these genetically modified plants, they are the plants whose gene has been altered by manipulation. Genetic modification of the plant is useful in different areas because of the following reasons. First, it increases the tolerance against many abiotic stresses such as cold, drought, salt, and heat. It reduces the it reduces the reliance on chemical pesticides, that we don't have to rely on these chemical pesticides because these crops are itself pest resistant crop. Now it reduce what? It can reduce the post harvest losses. It can increase the efficiency of minerals used by the plants. This prevents early exhaustion of fertility of soil. It can enhance the nutritional value of food. It could be like vitamin A enriched rice, which we are studying for so long, the golden rice. It creates the tailor made plant to supply alternative resources such as starch fuels and pharmaceuticals to industries. Next question says it is also a short answer type question, and this is NCERT Exemplar 3 question. This question is saying define antigen and antibody and name any two diagnostic kits based upon them. This is from NCERT session 10.2.3. So first, what is antigen? It is a foreign substance. It do what? It elicits, it triggers the immune response, which result in the formation of the antibody. Whereas this antibody, it is a protein and it is synthesized by the body in the response to antigen. So, what are the two diagnostic kit which is based on this antigen and antibody interactions? They are ELISA for HIV, which we have studied, and then the pregnancy test kits. They are also based on the antigen and antibody interaction. Next question. This is a short answer type question. And the question is asking us, how is a mature functional insulin hormone different from its prohormone form? This is from NCERT session 10.2.1, and this we have discussing in lot of questions earlier also. So what's this pro insulin? What it has? So we know that it has one extra peptide chain which we call C peptide or connecting peptide. This C peptide connects A and B chain in proinsulin. This is made up of 31 amino acid, whereas the mature function insulin is obtained by processing this prohormone. And in this process, what we do, we remove this C peptide and we mature this proinsulin to insulin, and then this A and B chains are subsequently get linked by which linkage? Disulfide linkage. So this is the picture. This has been shown that how this proinsulin has all three peptides, and then this green peptide, which is shown as the C peptide, has to remove, and then they have make a disulfide chain linkage between them, and this will make a mature insulin. Next question is also a short answer type question. The question says, transgenic animals are the animals in which a foreign gene is expressed. We know that the transgenic animals are the animals in which there has been the insertion of some foreign gene, and then foreign gene has been expressed in that animal. Such animals can be used to study the fundamental biological process phenomenon, as well as for producing products useful for mankind. Give one example for each type. This is from NCERT session 10.3. So we have studied like why these transgenic animals are useful. So we have studied that they are useful in the studying of normal physiology and development, study disease. Okay, then there has been the biological product formation, then there has been this vaccine safety, chemical safety, toxicity safety. So they are now asking us for one of the example. So what are these transgenic animals? So these are the animals in which a foreign gene have been expressed. Such animals can be used to study many fundamental biological processes. There has been a picture shown of transgenic mice, because we have studied that the 95% of the transgenic animals are what? They are mice. Now, for example, by using this model organism like transgenic mouse, how these genes are regulated can be determined, how they affect the normal body physiology and its development can be determined. Then we can study transgenic, we can study the chemical safety also, toxicity safety also. Then these transgenic animals can produce many useful biological products for mankind, as one of the example is transgenic cow Rosie, who can produce what? She can produce human protein, that is alpha lactalbumin, which is enriched in milk. They provide they gives us 2.4 gram of this protein per liter of its milk. So this milk is more nutritionally balanced product for human babies than a natural cow milk. Next, now there has been this long answer type question from NCERT Exemplar. The question says us, we have to define transgenic animals and explain in details any four areas where they can be utilized. It is kind of same of the from the last question. So we know that transgenic animals are the animals whose genetic material, whose DNA has been manipulated, altered to possess and express an extra gene. Following are the four areas where they can be utilized. First, as I told, they can utilize to study the normal function and normal physiology, function and development. We can study how the genes are regulated, how this can affect, those genes can affect the normal function of the body and its development. It can also help scientists to understand the normal gene expression at the various stages of growth and development. Then we can study about the disease of on this transgenic animal. So this transgenic animal can serve as model for human disease. We can insert some disease which is insert some gene which is responsible for causing a disease in human. They are designed to increase our understanding of how those genes contribute to the development of the disease. It make possible for the investigation of what new therapeutic, new treatments against that disease. The disease includes cancer, Parkinson disease, and all. Then we can use this transgenic animal for vaccine safety too. Testing the safety of vaccine before they are used on human is very important. Transgenic mice are being developed for this testing. Then we can use them for the chemical safety testing. Okay, by how we can make use them against this chemical safety testing? So we can make those animal more sensitive to certain chemical or drugs. So in a very short period of time now they are going to show us this toxicity or the side effect of those chemical and drugs. So the advantage is that we will get the result much faster, and this is how we can use it. Now the second long answer type question of NCERT Exemplar is that recombinant insulin. We have to list the advantages of recombinant insulin. This is from NCERT session 10.2.1. The advantages are there is no need of animals to be sacrificed for the production of recombinant insulin. Recombinant insulin was not found allergic to patients. The cost of recombinant insulin is not very high, the supply being many fold higher than the demand. Example is Humulin. We know that this has been produced first by Eli Lilly, which is an American company. Thank you.